So now we're going to continue this lesson with some more examples of basically what you can do with biotechnology and genetic engineering. Again, keep in mind this is just the sampling because there is a real whole lot of different, applications when it comes to genetic manipulation and biotechnology. Now, when we talk about genetic engineering, all of this that we're talking about is being able to cut and manipulate DNA. So remember we said nucleases are the enzymes that we use to cut DNA. And there are exonucleases and endonucleases. So if you think of what does exo mean, well, that means outside, right? So exonucleases will cut at the end of a sequence, okay? Whereas endonucleases will cut in the center, okay, within the sequence. So now, with these exo and endonucleases, keep in mind earlier I said that each of these nucleases will recognize a particular sequence, which is usually a palindromic sequence, and it will only cut at that particular sequence. And each enzyme will have a different sequence a lot of times. And like I said, if that sequence is only once in the DNA, then that enzyme will only cut once. If it's 5 times in that DNA, the enzyme will cut every single time it sees that sequence. Now, there are 2 terms I want you to know the difference between. So circle, star, highlight these because these are 2 of my favorite words in the English language. We have isoschismers and we have neoschismers. And both of these are terms for nucleases, but how they differ, okay? The trick that I use to remember which is which, I think of neoschismers first, because whenever you hear neo means new, okay? So new means that neoschismers are 2 enzymes that will recognize the same sequence of DNA, but will cut at different spots. Okay, so they recognize the same palindrome, but will cut at different spots. For instance, one may cut here, so look over here, one may cut the sequence after the G, whereas the other enzyme, the neoschismer of that first one, will cut after the A or after that A. So they both recognize the same sequence, GAATTC, but they cut at 2 different places. Okay? Now, isoschismers, on the other hand, it's a little confusing when I say this. They recognize the same sequence and cut at the same spot. So isoschismers would both recognize GAATTC, and they would both cut after the G. Okay? That would be isoschismers. Now you may ask, well, if they're both recognizing the same sequence and they're both cutting at the same spot, why did they have 2 different names, such as enzyme A and enzyme B? That makes no sense. Well, it makes sense in that they were extracted from different organisms. So let's say you found enzyme A out here on the, soil surface, and you found enzyme B out in an organism in the ocean. Well, yes, they do the same thing, but you name them different things because they're found in different places. And it's so important to know isoschismer and neoschismer because, for instance, if you're ever in, medical usage or research field, you run out of a particular enzyme. Well, if you know the isoschismer or the neoschismer, you may have access to it, right? Without having to delay your work or run out and order something new. Okay, so make sure you know the difference between isoschismer and neoschismer. If you're having trouble, just contact me on Remind and I will give you examples. Again, I will draw them out if you would like. The other point that I want to make about nucleases is that they'll cut the DNA either leaving a sticky end or a blunt end. And you can see that in this top figure here. So if an enzyme cuts straight down and there's no overhang, as you can see here, that's a blunt end. Whereas if it cuts like this, cuts after that G, cuts after that G, well, now you have that little bit of overhang when they separate. And those are considered sticky ends because think about it. This T is all by itself now, and it's going to want to find an A, okay, to stick to. So that's why they're sticky, because they like to grab a hold of other single strands. And sticky ends tend to work the best for research because, think about it, it's sticky, it wants to bind, okay? The last thing that we have here is modifying enzymes. And modifying enzymes basically means that these enzymes can add a methyl group, which you see in this figure here, and that methyl group actually prevents restriction cuts. Okay, so if you have methyl groups present, it will prevent cutting of that DNA at that particular location. The last thing that we have here is ligases, which you already know that term. Ligases are basically like glue for DNA. They covalently link pieces of DNA, so they're very valuable to manipulating DNA, because what good is cutting open that DNA if you don't have the glue to stick back certain pieces together? Okay. Again, if you have any questions about any of these terms we went over on this slide, just send me a remind message. Okay, so when we talk about genetic engineering, we also have what's called gene libraries and DNA libraries. Now, basically what this means is not only are we capable of cloning single or individual genes, but now we can clone libraries of whole genomes, okay? So as you see in this figure here, we can take an organism, let's say a bacteria's entire genome, so every gene it has, and what we do is we chop it up, and each piece ends up in a plasmid. And then we do transformation, and you get a single plate that has all of the genes of that bacteria's genome represented in that single plate. Okay, now keep in mind, each colony will not have every single gene. Okay, it's the whole plate that's the library. Okay, the plate will have all different bacteria, and each different colony will have different genes. So let's say this guy here has this first part. This guy here will have this second part. Okay, so each bacteria on that plate will have different genes, but the plate represents the whole genome. And since that plate represents the whole genome, you can then screen the plate to pick out whichever genes you want to work with. Now, there are also with gene libraries what we call metagenomic libraries. So they'll have a plate that has genes from many different bacteria. So a lot of times you see this, for instance, from an ocean environment. They'll collect a sample, and then they'll have a plate that represents all the genes in that particular location. They have prokaryotic gene libraries, which are, for instance, bacterial. They also have eukaryotic libraries. So these will have a lot of times yeast genes, or even you can do this with whichever organism you want. But basically the premise is the same, that the final plate will have the whole library represented. Then that brings you to the next point. Once you have this plate with all of the genes of an organism represented, well, what do you do with it? You have to screen it, right? Because now you want to know particular genes. Are certain genes there? Okay, where exactly in that plate are certain genes? So there are two techniques that we can use for screening a gene library. You can use hybridization, or you could use immunological screening. For hybridization, I want you to write down that this is for DNA. So you're using DNA probes, you're using single-stranded complements that are nice and sticky, and you're screening the library for the DNA, the actual genes. Immunological, you're now using antibodies to screen for gene products, okay, the proteins produced by the genes. Okay, so hybridization, you're going to be looking for DNA. Immunological, you'll look for proteins, particular proteins. Okay, so the first method is hybridization screening. Okay, I'm going to go through first, and then I'll give you the recap slide for you to take down your notes. So the first thing that you would do with hybridization screening is you would transfer your bacteria, which contain the fragments of the library, onto a filter. Okay, so transfer all of these bacteria from your plate that have all the genes present, and you transfer them onto a thin filter. You then lyse these bacteria, and you denature the DNA with alkaline or very basic solution. Okay? So now when you have denatured DNA, what do you know about single-stranded DNA? Well, single-stranded DNA will like to try and find its partner. Okay, it will want to stick to its partner. Okay, so what you do then, once you have this membrane of all this single-stranded DNA of this library, you then cover that filter with a solution that has radioactively labeled single-stranded DNA probes that complement the exact gene that you're looking for. Okay, you then develop that, and wherever you see the black, or in this case blue spots, that tells you that that's where your target gene is. Okay, that's how you find your target gene. So please remember that for hybridization screening, the key here is you take all of the bacteria of the library, you transfer it onto a membrane or filter, you break open those bacteria so it's just the DNA that you denature to make single-stranded, and then you target it with highly specific single-stranded probes that you can then detect. And those single-stranded probes will only bind to the target gene that you're looking for in that big library. Okay, so I'm going to give you a recap slide so that you can take down notes. Okay, so here you can pause the recording and write down the notes. The other way that you can screen a gene library is immunologically, meaning you use antibodies. So now it's the same kind of concept, but now looking for gene products, looking for particular proteins, because not only do you want to know the bacteria has the plasmid, but you want to know that it's functional. Okay, that you know which ones are actually producing the protein of interest. So what you do is, just like with the other one, you take those colonies from the plate, okay, and this time you're transferring to a membrane, and you're lysing them just like in the hybridization. So now you break open all the bacteria from the gene library, but now it's the proteins that you're targeting. So you use a different filter, a very particular membrane filter that the proteins will stick to. And the specific protein that you're looking for, you can then target with a primary antibody, because primary antibodies are specific to exact proteins that you would look for. You then apply the more general secondary antibody, which will bind to any protein, right? Any antibody. And that secondary antibody, what's important about that one is that it has detection on it. Okay, so it'll have some sort of probe or detection system on it Usually we use chemiluminescence. So basically you want to see fluorescence. It's kind of like thinking in terms of a Western blot where you get the fluorescent detection. So kind of think of this like screening for a particular protein, where it's almost like hybridization, but now instead of DNA, you're looking for a protein product. You take the gene library, you crack open all of those bacteria onto a membrane where their proteins will stick, and then you look for the exact protein that you want. And keep in mind through this, whether it's hybridization or immunological, you still have the original plate of bacteria. It's kind of like you made a copy of it, right? So once you find the location with the fluorescent detection, you know exactly which colony to go grab and allow to grow. Now, when we talk about biotechnology and genetic engineering, one of the most powerful techniques that you'll hear about is polymerase chain reaction, or PCR, which is a way to amplify DNA. Okay, so here we have PCR, which, like I just said, is polymerase chain reaction. Its purpose is to amplify DNA, and basically it makes a ton of copies of a very specific piece of DNA, whichever piece of DNA you're targeting. And you may ask yourself, well, why would you want to do that? What scenarios? Well, it's important in things like crime scene investigation, paternity testing, evolution, diagnostic medicine. So you use it a lot in hospital labs as well. Now, in order to perform a PCR, there are various things you need. The first one is the DNA template. So basically the target DNA that you want to amplify. Okay, you need forward and reverse primers, which are little pieces of single-stranded DNA that match the beginning and the end of the DNA sequence that you want to target. Okay, because as you know, polymerases can't start from scratch. They need something to have as starting points. Okay, you'll have the DNA polymerase itself, which is the enzyme that will build the copies of DNA. And it has to be temperature stable, because during this process you're going to, be using a machine that goes to high temperatures to denature the DNA, so the enzyme has to survive that. You need what are called dNTPs. Those are your nucleotides. So picture those as your A's, your T's, your G's, your C's, the bases that make up DNA. You then need buffers and magnesium chloride, because think about it, you need cofactors for that polymerase. The DNA polymerase is an enzyme, so you need cofactors to help it work, to activate it, and you need to maintain a good environment for this whole process. And the piece that students always forget, you need what's called a PCR machine. You need a very particular machine that will cycle through. It'll go to high temperature, cool down. It'll fluctuate temperatures for you for this process. Okay? Now, with that fluctuating temperatures, the reason that is that each PCR, there are 3 steps to happen. You need the machine to first denature the double-stranded DNA, which happens at high temperatures. So you see that here. You heat, the machine will go to a high temperature to separate the double strands of DNA. It will then cool down so that the short primers targeting your specific sequence can bind. Because think about it, if the machine did not cool down, what would happen? These guys would denature too. They wouldn't be able to stick. So it goes from hot to cool, and then it goes to the temperature for Taq polymerase or whichever DNA polymerase you use, because each enzyme has an optimal temperature. Okay, so the first step is high temperature to separate the double strands so that you can then cool the temperature to allow the primers to stick, and then go to the optimal DNA polymerase temperature, such as 72 degrees Celsius, for the polymerase to then build from that primer, okay? And it makes copies of that DNA sequence. Now, you don't have to be able to draw all of this out, but this is just to show you that you don't get your target DNA until 3 cycles later, because each time those primers are, building the polymerase is building from the primers, you end up with a lot of overhang sequences. And as you can see here, this is when you get the nice target DNA. Okay, so you don't have to be able to draw this out or anything. Just remember the fact that it's not until 3 cycles of PCR that you finally get the properly sized target. Okay, circle, star, highlight, 3 cycles. Now, when we talk about PCR, there's a lot of different types of PCR that, basically they have made this technique more and more powerful for us researchers. One of the ones that we use a lot is called qPCR, or quantitative real-time PCR with TaqMan probes. Okay, what this means is not only can we amplify or make a ton of copies of the DNA, but we can quantify it as well. So this one will quantify while it amplifies because it's using these special TaqMan probes. And what these TaqMan probes are, you have to know what they are. They have a fluorophore on one side, so a fluorescent probe on one side, and then what's called a quencher on the other side. A quencher, like when you quench your thirst, it blocks the fluorophore, okay, because right now it's connected to that guy. So when the quencher is near the fluorophore on this TaqMan probe, the quencher blocks fluorescence. What then happens is this TaqMan probe, it's a single-stranded piece of DNA that binds in the PCR to the target DNA that's getting amplified. What happens when the polymerase is amplifying that DNA? Taq polymerases have what's called exonuclease abilities. So that Taq polymerase, as it's building the new strand, it will then come along. Once it makes it to this probe, it'll chop it up. So when the Taq polymerase here chops up that probe as it's building the copy of DNA, what happens? The fluorophore is now floating away. It's separate from the quencher. What happens when the fluorophore is separate from the quencher? It fluoresces. Okay? So the more initial product that you have, the more template DNA you have, the more new strands will be built, the more probes will get cleaved, and the more signal you'll see. So that allows us to quantify it. Okay, so one more time. The more product you have, the more template DNA you have, the more new strands will be built with the Taq polymerase, which means more probes get cleaved, which means greater fluorescence as the fluorophore is separated from the quencher. Okay, this is a slide that helps you kind of see in words what I was just saying. Make sure you're very comfortable with knowing what happens with the fluorophore when it's near the quencher versus when it's separate. Okay, make sure you can explain how exactly does a TaqMan probe allow us to quantify a PCR product. Okay. Similar to that, sometimes you will hear reverse transcriptase PCR. What reverse transcriptase PCR is now you're amplifying genes using mRNA as the starting point, because as you know, reverse transcriptase means you're going from RNA to DNA. Now, why would you want to use mRNA as a starting point of your PCR? Well, this is used in gene expression research. okay, because mRNA is gene product, so it shows how active particular genes are, okay? So you're now going to have to clone copies of intron-free DNA in this process and amplify that. So the main steps of this, of a reverse transcriptase PCR, first you have to isolate that RNA, the mRNA. You then clean it up to get rid of any DNA that might be contaminating it. And then you do what's called first-strand cDNA synthesis, and you do not have to memorize these steps of first-strand synthesis. It's basically meaning that the machine will make the complementary DNA copy of that RNA, okay? And then once you have the DNA copy of that RNA, now you can put it into the PCR and amplify it, okay? Because PCR only works for DNA. PCR can only amplify DNA. So if you want to study RNA with PCR, you first have to make a DNA copy. So that's why you need reverse transcriptase PCR, okay? Keep that in mind, circle, write down that note, put little stars next to it. PCR only amplifies DNA, okay? Now that reverse transcriptase PCR is very valuable to us in the research field because it allows us, like I said, to examine gene expression, okay? So you can look at whether a gene's expression is upregulated or downregulated in certain conditions. Basically meaning you'll have two different conditions and you'll ask yourself, when do I see a greater signal? When do I have more of that fluorescent final product? So if you see a lot of reverse transcriptase PCR product, well, that means that there was a lot of mRNA present to turn into the DNA and make copies of it, right? So that means that gene expression was high in that condition. Whereas if you do not see much fluorescence or amplification at the end of the reverse transcriptase PCR, well, that tells you there wasn't DNA to copy or amplify because there wasn't mRNA to copy, which means the gene was not being expressed. Okay, so reverse transcriptase PCR lets us look at how much gene expression there is in different conditions. Okay, so it's a very powerful technique. There are also a lot of medical applications of PCR, whether you're doing regular PCR or the big time qPCRs, okay? And with these, bullet points that I have on this slide, keep in mind that there's a whole lot more you can do with PCR. This is just the sampling. It's very powerful technique because you're able to amplify or make copies of very small amounts of DNA, which if you think about it, well, that's great in forensics, especially since at a lot of crime scenes, you're not getting a lot of DNA or it's been highly contaminated. So this allows you to kind of amplify your sample to work with. It's also very powerful in diagnostics because we know the genomes of almost every bacteria or virus that are most commonly infecting people. So whenever we find new viruses, new bacteria, first thing we want to try and do is, get its genome, sequence its genome. And then once you can do that, you can use PCR to see does a patient have it or not. For instance, you use primers that target sequences found in that virus or that bacteria, and you test your patient's blood samples or swabs or pus samples, anything like that, just to see, okay, if this person's infected, the PCR primers will pick up the viral or pathogen genes and make copies of it. And you'll see those copies on a DNA gel, like on the bottom of this slide here. Okay, that's DNA gel. You can see bands when the amplification occurred. Now, if that patient's not infected, the primers won't find anything. They won't have anything to bind to. There won't be any amplification, and you won't see any band on the final gel. Okay, and they do this for a lot of different infections, and they even look at, mutated genes like BRCA1 for breast cancer. They'll have primers that target the mutated sequence and see if that's present in a sample. If you see amplification, that means the gene you're looking for is present in that person. And if you don't see amplification, then the primers had nothing to stick to. There was no bad gene present. Okay, so very powerful in the medical field. It's also very powerful in the environmental field. Okay, environmental field, I'm not going to go into too much detail here, but I basically wrote out a lot on this slide just in case you're more curious about how it's actually used environmentally. But basically, you're able to target bacterial genes to see if they're contaminating certain areas, or to find particular metabolic aspects that might be valuable from different bacteria. So for instance, if there's a hot spring, these very crazy conditions that can withstand heat, well, let's gather the microbes there and amplify their genes, see if there's a product that we can use. Okay, so we're able to find very valuable enzymes for our research, even things like the PCR polymerases that we use. We have gotten from, trawling ecological samples, looking for amplified gene sequences. And then the last one here that we have is the idea of rescuing DNA from extinct life forms with PCR. Now, this is kind of like the Jurassic Park type of scenario. It's not completely, realistic in some senses, because cloning such large amount of genomes can get problematic. A lot of times the vectors can't handle it, or you have a lot of mutation issues or instability issues. But just be aware that trying to rescue old forms of DNA is one other possible use of PCR, because basically once you look at sequences, you have sequences, if you can make little primers for them, you could amplify or target any particular genes you want. Okay, so we're not going to go into crazy detail. I just want you to know that there are environmental and extinct life form uses for PCR. The last few slides I want to cover are the molecular techniques for analyzing DNA. So with blotting, there are a few different forms of blotting that I want you to know the difference between. Now, in all of them, you'll have fragments run on some sort of a gel, then copied onto a porous membrane, and then probed to have some sort of detection. Now, what I want you to know is the difference among southern blot, northern blot, and western blot. And some students like to use the trick of snowdrop to remember that southern blot is for probing or detecting DNA, northern blot is for detecting RNA, and western blot is for looking at proteins. Okay, so in all of these, you're putting fragments onto a membrane, but in southern blot, you're probing for DNA, in northern blot, you're probing for RNA, in western blot, you're probing for proteins. Okay, and it's more specific blotting than simply running a gel electrophoresis. Now, gel electrophoresis is one of the most commonly used techniques. What you basically do is you put DNA fragments into an agarose gel, and that agarose has a network of varying size pores. you put in a salt buffer, and if you think about it, salt conducts electric currents. So you apply an electric current, and as you already know, DNA has a negative charge. So when you put that electric current through, those negative charges are going to flow toward the positive side of that tray that you have set up. Okay. Now, in addition to your DNA samples, you also load a marker or a ladder, and that's important because it will have known size fragments of DNA that you can then compare your sample to and figure out exactly what size DNA you have in your sample. Now, with this concept, you have the negative DNA moving its way, navigating, snaking through the pores of that agarose gel, trying to make it to the positive side of that gel. And be careful, because it is possible for your DNA to flow right through, it's so desperate to get to the positive electrode that if you leave it too long running, your DNA can run right off of the gel into the salt buffer. Now, the way this gel concept works is smaller and larger fragments will move slightly differently. So think about it. These DNA fragments, they have to navigate, snake their way through the pores of this gel. If they're bigger fragments, it's kind of like when you see adults in a children's playground and they're running after their child. And sometimes the adult, because they're big, the playground's not meant for their size, they'll get stuck, trying to navigate through. How many times have you seen on YouTube, some big parent getting stuck on a slide or something, those tube-like slides? So that's what happens with larger fragments. They get stuck early on in the gel. Okay, so they'll stay very close to where you loaded them into those wells. Whereas the smaller ones, they can zigzag just like little kids running through the pores, and they make it farther. Okay, so larger fragments are closer to the wells where you put them, and smaller fragments of DNA on a gel will run further. The last important thing when you're doing gel electrophoresis is to remember to have ethidium bromide. And ethidium bromide has two functions in gel electrophoresis that I want you to know. One, it's what we call an intercalating agent, INTERCALATING, intercalating agent. That means that it pools between or collects between the base pairs of DNA. The second characteristic is that it glows under UV light. So when you put these characteristics together, it means that when you have ethidium bromide in your gel, it will only end up located where the DNA is because it pools or collects in the DNA bases. Then when you shine UV light on this gel, it'll fluoresce, it'll glow for you. So it'll show you exactly where the DNA is. So this is just a visual to help you see. Again, you load your DNA into wells on the negative side of the tray, and then these smaller fragments will run further. They'll be further away, trying to make it to the positive side, whereas the ones closer to the wells will be larger sizes of DNA. And then you use UV to visualize. And why does that allow you to visualize? Because of ethidium bromide. Now, in addition to visualizing DNA in terms of the gels, you're also, when you do research, able to figure out the concentrations of your DNA and RNA. And what's really important here is because in a lot of our experiments, when we try and isolate DNA or RNA, a lot of times the samples can get contaminated. And it's natural. There's so many proteins everywhere around you that when you're doing these experiments, you can be very careful, and still your sample, even from the cells that you're cracking open, sometimes you try and clean up everything, but things happen. And so you're able to use UV spec or UV spectrophotometry. Sorry, I mess this up every time, even though I've used it for over 15 years now. But yeah, spectrophotometers or spectrophotometry, that's why I just say UV spec. You can use UV spec to kind of see how pure is your sample. And I want you to be able to tell me, basically in questions like on an exam, to be able to tell me if I give you what's called a UV spec ratio, 260 over 280, you should be able to tell me how pure of a sample, whether it's pure DNA, whether it's RNA contaminated or protein. So basically the concept is DNA and RNA absorb UV light at 260 nanometers. Okay, so think of 260 as DNA and RNA, whereas 280 on a UV spec represents proteins. So when you then do the ratio of 260 over 280, pure DNA will have a ratio of 1.8. Circle, star, highlight that number. 1.8 on a UV 260 over 280 tells you you have pure DNA in your sample. You did a beautiful job of isolating DNA. If protein is present, well, what did we say protein is? 280, which is the bottom of our fraction. So if protein's present, you just increased the bottom of a fraction. So for instance, you now went from 1 over 2, 1/2, to 1 over 4. What do you know about 1 over 2 versus 1 over 4 now? Well, 1 over 4 is a smaller number, right? The bigger the bottom of that fraction gets, the smaller your ratio is. So if you end up with a UV 260 over 280 that is less than 1.8, you have a lot of protein present. So you'll usually see it as like a 1.3. So if I ask you, what does it mean if your DNA sample has a 260 over 280 ratio of 1.3, well, it tells you that there's a lot of protein contamination. Okay, make sure to write that down. If RNA is what's actually contaminating your sample, then instead, this top number gets bigger, okay, because RNA, the difference in terms of uracil and the single-stranded nature, it ends up having the pure ratio of 2.0. So if you see 1.8, your sample is good and pure DNA. If you see something like 2.0 or 1.9, you have RNA contamination. If you see something like 1.6 or 1.3, you have protein contamination. Okay, make sure you're comfortable with those 3 scenarios. If you're unsure about this, just send me a message in the Remind app and I'll give you the exact format of how to practice questions like this. The last thing that I want to do here is just to briefly mention the idea of applications of biotechnology and the kind of ethics that you encounter, okay, because it's a big thing today, you know, especially with the media kind of blowing things out of proportion and social media existing. Biotechnology gets a lot of bad rap, and it's mainly because people don't necessarily understand the science behind it. Now, biotechnology, the exact definition is when you're using living organisms to create a product or to kind of modify products to help humans or other organisms, okay? So the idea behind it is to be helpful, but a lot of people are scared about it. Genetically engineering organisms has been used a lot of different ways, biologically, pharmaceutically, producing things like insulin, which is very valuable to people. When you do these things, such as you kind of see on the bottom here, genetically engineering corn to what we know it today, fish to increase size, for instance, or increase the amount of fruit, or to even help produce things like, in this case, immune response. So rather than having old school vaccines, but giving third world countries fruit that trigger immune response. All of these things, they're meant to kind of use genetics and biotechnology to help people. It's using what we talked about in this lecture to introduce new genes, which introduce new proteins. Okay, so once you do this, those organisms are now called transgenic because they have new genes put into them. It's used with things like vaccination, crops, medical, like I said, things like insulin. Okay, a lot of times the things that we're trying to do with genetic engineering, especially in agriculture, it was to make crops more robust. So produce more fruit, like I said earlier, produce bigger fruits and vegetables, have healthier plants, bigger size, resistant to things. They do this with animals to make them bigger, more meat, more, taste, enhance the products. And like I said, medically, things like, producing increased amounts of insulin or various hormones or gene products that we need. Okay, so you can kind of look into it more, make your own decision whether you're for or against, but the way I see it is it's done a lot of good. Now, there are concerns from people because they think that it's too new to kind of see the full implications, and they worry that there may be bad side effects down the line. But at this point, we have had quite a few generations to look at these things. But, time will tell. So always research, look into things from reputable sources, truly vetted, peer-reviewed journal articles, rather than listening to the media. Don't let social media, don't let news media make the decisions for you, okay? Because even as you see with things like coronavirus or E. coli outbreaks, if you don't know the science and you don't look it up, you can start believing the scare tactics and the false reports that they make about things like those events or vaccines or GMOs. But be your own advocate and really look into these things, because you'll see that a lot of the so-called bad things that they try and say about things of science, not true. Okay? So this is just a touch upon the subject. If you're more interested, make sure to look more into biotechnology, because it's a huge field now. It has a lot of potential value. Okay, that is it for this lesson. As always, once you get to the end of a lesson, make sure you are heading to the discussion board, and also please make sure to add this lesson to your current study guide that you're building. As always, if you have any questions or concerns, please contact me in the Remind app. Thank you and have a great day.